Hello, I'm Art Modazenik, and I represent the American Association of Pharmaceutical Scientists group and task force that is putting together a series of distinguished pharmaceutical scientists lecture series and interviews. Our program is promoted by the Distance Learning Committee of the AAPS. It's my pleasure this morning and this afternoon to welcome Professor Joseph Robinson of the University of Wisconsin as our distinguished pharmaceutical scientist. Joe, I'm happy to welcome you to our series.
Thank you,
Look forward to interviewing you.
Good, thank you.
One of the pleasures I have in this program is to invite your motivation for becoming a pharmaceutical scientist. I'd like to hear a little bit about how you were born and raised and what motivated you, maybe from an educational point of view, maybe from a career point of view, to go into the pharmaceutical area or even to science in general. And were there any other disciplines that you favored at the time that you were coming out of school, let's say at high school level and so forth?
Well, I guess I represent the post-Second World War generation which had followed on the Great Depression. And so a career, a job, education were dominant in the family. I was rather fortunate as a young man, and I was very young. I couldn't have been more than 7 or 8 years old, to start as a delivery boy in the local pharmacy. I suspect that most people did something akin to that. But it was, I graduated from delivery boy to waiting on customers to eventually practicing as a pharmacist for a year in that same pharmacy.
And that was in what community?
That was in New York City.
Mm-hmm. And what borough?
Washington Heights and Manhattan.
Yes.
My colleague George Lagraffi lived about 4 or 5 blocks from me. Mm-hmm. Although we didn't know each other and he was a few years older than me, we nevertheless share similar cultural background. But at any rate, when it came time for developing a profession, I never thought about any other profession except to become a pharmacist. I thought that was the end of the world for me. And so I began at Columbia University. There were, at the time, 3 or 4 schools in the New York area. But Columbia had a certain attraction, mostly because my mentor in the pharmacy had gone to Columbia many decades before. But it was a very interesting school, very technology-oriented, and very little science at the time. But the science developed over time. George, upon receipt of his PhD, came back and taught at Columbia for a while. And so I saw him his first year out of school. I was still a graduate student at Columbia. And while there, I had interesting influence by a couple of the faculty members, and it was done in a rather offhanded way. One was a pharmacologist by the name of Michael Clay, who saw me in the cafeteria and he said, "Did you ever think about graduate school?" And I had never thought I was capable of going to graduate school. And the other was a faculty member by the name of Joe Koenig. And Joe also suggested I go to graduate school. And so I applied to the better schools that I knew of, which included Wisconsin and Michigan as the top 2 contenders. And I wound up coming to Wisconsin. I didn't know anything about it. I thought there was wilderness out here and Indians still on the plains, but I came out anyway. And best decision I ever made in my life. But that's another story. But that's a little of the primer of how I got to wind up at Wisconsin.
Was your trip to Wisconsin to look the campus over and the school over, was that your first trip essentially out of the greater New York area by any chance?
Absolutely.
It was for me as well. I was right across the river from you at Fordham at the same time you were in Columbia, and I had the same experience that when I came to Wisconsin, that was the first time I ever came here. So you were a pioneer and a visionary like the rest of us. The Eastern schools produced a lot of graduates at the BS/MS level that eventually came to Wisconsin for PhDs.
Absolutely. But I recall my mother saying to me, "You don't want to go out there. There are violent people out there." She, of course, hadn't traveled beyond the Hudson River either.
And so you also took time enough, though, at Columbia to earn your master's degree while you were there. Who did you study with at?
Joe Koenig.
Joe Koenig as well, yeah.
That was my primer. I really wasn't confident enough to know whether I could do that, whatever he was talking about. Remember, the curriculum at Columbia was largely technology. And I thought, well, what am I going to do with this? And can I do it? And so the master's was a way of convincing me that I could do it. We took a lot of courses at the uptown campus of Columbia. And
in Morningside Heights?
Yeah, Morningside Heights. And that gave a little stability to the master's program and raised it from technology to some science. But after the master's, it was clear I needed to go somewhere where science was practiced big time. Mm-hmm. And that led me to Wisconsin.
Had you had any exposure during your undergraduate or master's program with industry at all? Did you have any internships, or did you visit companies that kind of gave you a flavor of the kind of research or development work that was going on in industry?
Well, actually, I had spent a summer at Bristol-Myers, and it was sort of an orientation towards industry, but not a very good one. But some of the faculty took us to hear speakers from industry, and I got excited about what they were doing in industry. Mm-hmm. And I thought, this is a field that is ready to move from where we are to something else. I have to tell you a funny story. Both George Zagraffi and I, at our undergraduate level program, were exposed to the transition in pharmacy. We went from a lot of natural products with extracts and powdered extracts and liquid extracts and syrups and tinctures, and so our curriculum was loaded with memorization of common names and trivial names, and that stuff is almost never used today. So because it wasn't really science-based, we became outmoded in a very short period of time. But that was the essence of the curriculum in those mm-hmm. days. And we were in the transition years that where they were moving to solid pharmaceuticals away from compounding, and it was an interesting time.
So you were attracted to come to Wisconsin. You said you had considered Michigan as well. And when you came to Wisconsin, did you have a preset idea of what kind of a post-PhD career you're going to have? Were you already thinking academic, or were you interested in everything?
I actually never thought about an academic career my entire time at University of Wisconsin. I had done a little inquiry, and I had heard about my mentor, Takeru Higuchi. And I came out here with the idea that I would work for him. And I was fortunate. He took me on as a graduate student. But I never thought of an academic career until the associate dean at Wisconsin in the 60s, Louis Bussey, came up to me one day and said, have you ever thought about an academic career? And I said, no. And he said, well, we'd like to see you stay here. And I was very flattered. And so I asked a colleague of mine who became a very important part of my career, Ken Connors, for advice. What did he think about it? Well, in those days, the school was run in a fairly autocratic way. He didn't know anything about it. Mm-hmm. So anyway, I accepted that position. I had interviewed at Merck and SmithKline and had offers from them, but I took the position at Wisconsin. There's a funny story with that, Art. When Louis Bussey offered me the position, he didn't tell me the salary. And I never asked the salary because I thought he might withdraw the offer if I seemed too eager. So I didn't know how much I would make until I got my first salary check. And it was a whopping $9,000 a year. And I was in hog heaven compared to earlier salaries. So it was an interesting period of time.
I remember one of the folklore stories they tell about you, which is, I think, a compliment to you, is that amongst the graduate student community, you were tremendously approachable by the foreign students. They like to appreciate the time you took with them, not just to explain the scientific question they had, but to maybe ferret out the cultural problems they were having in adapting to the language or the style or personality of their instructor or classroom. That seemed to me to be sort of the hallmarks of a good teacher right there, even amongst your graduate students. And that lesson bore out in the fact that a lot of those graduate students came home to be some of your closest friends and associates later on in your career. Would you elaborate on that?
Well, when I came to Wisconsin, I didn't consider myself very well educated, certainly not culturally educated. And I found it absolutely fascinating, and it was more for my benefit than theirs, that I talked to foreign students, and I was fascinated about issues that they were confronting, both politically and religion-wise, and almost every way imaginable. And I learned a lot from the students. I learned more from them than I gave to them. And in a similar sense, I didn't find I was terribly interested in world events. So it's at that time that I started reading newspapers and magazines, and I don't know, but it became sort of a terrible urge for me to become well-rounded, educated, and know what's going on in my surroundings. So maybe that was the reason, Art, but it was from a position of weakness that I did that. It was to learn more about other cultures and what's going on in the world.
One of the challenges that a young professor or academician has in creating a career path, especially in an institution where your major professor may still be on the faculty, is to create a distinguished mark of your own. How did you go about charting a course for your own individual sphere of interest in science, and what were some of those disciplines? How did you migrate from your, let's say, your dissertational interest to the early research interest you had while a graduate student to that of a young faculty member?
Well, first I should tell you that my major professor, mentor, Takeru Higuchi, didn't like biology. And when I started in biological work, he took me off his Christmas card list. And I was off that for 4 or 5 years. And one day he came, I saw him in the hall, and he said, I saw an article you published, very interesting. He didn't say it was good. Interesting. That's as strong as he would get with biological sciences. But after that, I got back on his Christmas list. Mm-hmm. And those were always informational missiles that told a little bit about the family and what other people in his group were doing. So I felt pretty good. But actually, the people who were instrumental for me in this were 2 colleagues that I consider to be very important in my whole career. One was an analytical individual by the name of Ken Connors. He's retired now. And Ken was very active in what was referred to as physical organic chemistry and mechanisms in that. And he had spent most of his research career studying complexation. And I saw what he was doing, and I thought, this is a dead-end activity. So he taught me and convinced me not to pursue that area. Another individual that was very important for me was Stu Erickson. Stu Erickson was a colleague, a faculty member at Wisconsin for a few years, overlapped with me, and when I was a graduate student, and left and took an executive position at Allergan, a prominent ophthalmic company. And he used to talk to me about how little was known about drug delivery to the eye. And we talked at considerable length. So I embarked on what I thought would be a systematic study of the eye, always looking for physical chemical understanding coupled with physiological events in the eye, such as tear flow and why drugs are removed from the front of the eye quickly because of tear flow and tear turnover. And I expected to do a lifelong study over the next 30 or 40 years with first studying the front of the eye and then the anterior chamber and then the posterior chamber. Well, I never got much beyond the tear film, and so I spent most of my career and in fact still have research going on in the eye. So that was the start, and I had my first NIH grant from the National Eye Institute, and that was a great event for me, although getting money from NIH was a lot easier in those days than it is today. But that was the beginning for me, and then I evolved into other areas, mostly because I didn't want to be seen as one-dimensional, and I thought I needed to expand. I still had that crazy generalist feeling that young faculty often have, and it infected me as well. But that was the beginning of my career, and very enjoyable. And I'd like to think I made a contribution in that area, although after a period of time, your early work is never referenced, and that's okay. I've accepted that.
Well, we'll talk about some of these other areas of biology in a moment, but I wanted to just dwell a little bit in depth about your contributions in ophthalmic drug delivery and ocular systems in particular, because your legacy, Joe, is not just the direct contribution you've made, but the fact that probably some of the 5 or 6 most prominent ophthalmic drug delivery scientists in the U.S. and in fact in the world today are either direct students of yours or people who are much influenced by your critical contributions in that field. And I know that you've been recognized in the Control Release Society and in drug delivery journals and so forth in this area. This must be a degree of satisfaction to see this field expand beyond the challenge that Stu Erickson gave to you, because your approach to the whole pharmaceutics approach to drug delivery, we'll talk later about how you extended it to other fields, in ophthalmic persisted. You stayed in that field for decades.
Yeah.
Could you comment about the legacy of continued interest because of your students in this area as well?
Yeah, I was very proud of the fact that a number of my students chose academic life, in which case they were free to pursue lots of different things if they so chose, and many did choose to pursue ophthalmic studies. And a number actually started working at Alcon and Allergan and the prominent ophthalmic companies. It was a little difficult at first because they were competing with me at NIH and other places, but I became very proud of their success, and I was pleased that they took this well beyond what I ever could in my lifetime, and took it to distant areas, which includes gene therapy and a lot of enzyme work and carrier transport systems, and very fine work. And they're still pursuing it. And in fact, I have grandchildren academic grandchildren that are pursuing this as well. So
Students of your students.
students of my students.
Yes.
And that's always fascinating when I run into these. And sometimes it's, well, I don't keep track of who has worked for what faculty member, but often at a scientific meeting, somebody will come up to me and say, I'm your academic grandson.
Well, I can think of a direct correlation there because of the ophthalmic contributions of someone like Ashim Mitra comes because he's a progeny of one of your graduate students. He came out of the Patton and Michelson and Kansas group. And I remember, Joe, if you recall, when you came to visit us at Merck as a consultant, that we had a very strong ophthalmic drug delivery initiative because Merck stumbled into timolol maleate as the top ophthalmic product in the world. And we happened to have the Robinson School of Thought there. We had Michelson and Patton on the Kansas faculty, and you were very prominent. That was in the 80s, and to this day, that's continued. And as you said, you're the grandchildren in the discipline. That must give you a great source of satisfaction to see that continuity.
Oh, it absolutely does. And not so much the continuity, but the fact that the franchise, which is ophthalmic drug delivery, has been and will continue to be studied in a serious way from a drug delivery point of view. I was very pleased with that. You probably are aware, Art, that the Eye Institute, which funded a lot of the work, was one of the newer institutes. And having an orientation towards drug delivery in a field that had otherwise been surgical and non-drug related was important. And it remains that way to this day. So.
We don't always do this. I'd like to often ask this question toward the later stages of the interview, but since we're on the ophthalmic, would you express a vision where you see maybe the outreach in ophthalmic drug delivery going in the 21st century? Because you're probably most qualified to offer that kind of an opinion.
Well, I can give you a sense. I would quote Bob Langer, a colleague at MIT, who was asked what we should expect to see in this century. And he said, elimination of disease. That's a mighty undertaking. But his view was that once we can begin to manipulate genes and understand what's causing disease, we should be able to do away with a lot of these diseases. I look at the ophthalmic world, and ophthalmology is a very difficult area because putting drugs on the front of the eye to treat infections and fungal issues, etc., is pretty straightforward. The drug is reaching the target immediately. But if I want to get to the back of the eye to treat retinal diseases, and a variety of other diseases which are now prominent in the elderly population, I don't really have a way to get it back there. There's a blood-brain barrier, so the drugs that you give systemically don't reach it. So there's a significant challenge that is now dominating a lot of the ophthalmic world, which is how do you treat these diseases in the back of the eye in an effective way? And there's a search for a different type of delivery system than had ever existed before. It's not solved, but it will, and that's one of the challenges that's coming up. But in the broader sphere, I suspect that there will be a large influence on genetic issues and gene-related diseases. The eye is accessible. It's not easy to deal with because you can't poke human eyes as you can to draw blood from humans. So it's a little more difficult in that sense. But it is, if I ask you on a personal basis to identify the sense that you'd least like to lose, it would probably be vision. You could tolerate losing hearing and maybe touch and taste and the rest, but vision is one that you don't want to lose. So there'll always be an active interest in ophthalmic research. From a drug delivery point of view, there are enormous challenges that are currently being studied and will be studied for the next 2 decades, I'm sure.
Well, I'm sure you're right about the sense of sensual perception and the importance of vision, and also quite obviously the concept that with increased number of aging folks in our population and vision being a bigger issue in health, that ophthalmic drug delivery will continue to play an important area in frontier work. Yes.
Yeah, absolutely.
Oh.
Yeah.
Well, thank you for those insights. One of the things I'd like to explore with you, which is very exciting, is the fact that one of the important contributions that you've made is that you've taken the seeds of observation and analytical insight into a scientific area in one discipline and used it and converted it to another. So that when you looked at ocular drug delivery, you thought about drug delivery and getting to the retina and looking at the loss of delivery, and you looked at controlled drug delivery. Joe, you made a very important contribution in editing some very important books in which you consolidated the advanced thinking in controlled drug delivery and became probably the most recognized scientist in the world. In fact, I think you were identified in a poll as the most recognizable pharmaceutical scientist in the 80s because of your contributions in controlled drug delivery, largely because of the fact that you edited and correlated and pooled these scientists together. How did you go about getting into that field where you broadened your area from ocular and used those seminal ideas that you talked about, pharmaceutics and physiology, and took it to whole drug delivery, controlled drug delivery?
Well, there were a couple of issues that I think were seminal in that. I had mentioned a colleague, Stuart Erickson, who was a very instrumental individual in my life. I had actually started to work with Stu while I was a student with TAC on modeling using an old analog computer with wires and the like sticking out of them. And we had actually followed up on a study that was done by a prominent scientist in the UK, part of his thesis work. And this modeling looked at parameters such as flow rate, issues for the oral route. There's a whole bunch of things, none of which we understood very well, if at all. And so part of my reasons for doing this had to do with the genesis of whole fields. Pharmacokinetics was an issue that was becoming prominent. Some very good scientists in the industry and in academia were pursuing in North America the frontiers of pharmacokinetics. That, of course, led itself to understanding the parameters that would go into that pharmacokinetic modeling. And so the timing was correct for that. The larger sense was that I saw a need for applying physical-chemical principles to drug delivery. That meant in the eye, when I looked at flow rate, tear turnover, and a variety of physiological issues, that I then should begin to build into models what all of this meant. You may recall, Art, that a very good study was done looking at a cancer drug in which biological parameters were examined. It came out of the NIH, NCI actually. It was never really followed up extensively, but it was a beautiful study. And that was in and around the time when I was starting to look at other routes and other forms of drug delivery. And I thought that we had too many missing pieces of information. And if we filled in some of the blanks on that, we could do drug delivery in a much more sophisticated manner. And that happened. It didn't happen on my watch. It happened by a variety of scientists who began to study models, animal models, and tried to relate those animal models back to humans. And we started filling in the blanks on a lot of issues that were very important in drug delivery. So I think that was the genesis of it.
So that analog computer brought you into looking at pharmacokinetics applied to the disposition of drug release.
Right.
You played a prominent role in meetings associated with controlled drug release.
Right.
Were you present at that Birmingham meeting where the Controlled Release Society was founded? I thought you were.
No, it predated me, actually.
Okay.
I did serve as president of the Controlled Release Society, but it was much later. And the organization has been still reasonably small. It hasn't grown a lot over the years, and I'm not quite sure why. The meetings are quite good, actually, but it's been a small boutique organization.
But the discipline of control release became identified. When I mentioned that you had been identified as one of the most cited and quoted scientists, that data came to me from Eugene Garfield, who was the editor of Current Contents. He and I were on a panel together in Louisville, and he asked me about Joe Robinson, who was the most frequently quoted control release scientist. And I said, well, he's drills down very deeply in so many areas, but he has command of the whole field. So I knew that you were involved in the organizational efforts to put that as a fundamental discipline.
Yeah, I was rather fortunate because we could put on programs, short courses and the like at the Controlled Release Society. And my successor at Controlled Release Society as president was one of my former students, Vince Lee, who kept at this. And we tried to make it a global organization. So we alternated meetings between the Far East, Europe, and the United States. So every year we were in one of those three locations. And I think it helped to globalize the field of controlled release. I'd like to think it did anyway.
And it took you outside of the formal pharmaceutical disciplines, so to speak, as a specialization into areas of a broader outlook in multidiscipline science as well, didn't it?
Oh, yes, absolutely. In fact, I think it's fair to say that my interest in biomaterials, as evidenced by the bioadhesion work, came about because of the world of polymers. And so I interacted very extensively with the polymer people, and I found that we were both chasing the same type of endpoints in products and issues that we thought were important. And they were very successful at getting money from the surgery study section at NIH, and I was always amazed at how individuals trained in biomaterials and in polymer work were just outdistancing the pharmaceutical scientists. There's just no comparison. So I thought it would be a good train to get on, and so I incorporated a lot of the world of biomaterials and polymer work into my way of thinking.
You've recollected your relationship with some of the mentors that Erickson and Higuchi and Koenig had in your life. And now you've talked about the outreach you made toward international communities. Incidentally, I remember being with you and Vince Lee in Amsterdam on a program together where you made a particularly strong appeal to the European community for some of the work you were doing in bioadhesives at that time, which we'll get to in a minute or so. But your colleagues, now I'm talking about not the mentors or your students, but the peer level relationships. Who were some of the people that you might have collaborated with over the years that were most influential in maybe satisfying your scientific interests, or maybe even challenging you or supporting your viewpoints and disciplines?
Well, I was very lucky, as you know, Art. When I first went to Wisconsin, Takaguchi had a rather large group of people. You were one of them. And many of those people wound up in academic life. And they all had a commonality of a basic science orientation rather than the technology that has dominated this field for a very long time. And so at an early stage in my career, I was introduced through seminars, through contact, through discussions, endless discussions with other graduate students in TAC's program about what should be done in research. And it had a very large influence on me because it dominated, and especially when Takaguchi gave me a rejection on the biological work I was doing, that was a further incentive to try and prove him wrong, that you could do good work in biology, but you just had to control more of the animal studies that you were doing. And so I count as very influential most of the graduate students that I had contact with. There was a culture established at Wisconsin that I never forgot, that I'm grateful for even to this day. And of course, at the same time, I would say that my family was instrumental in allowing me to advance my career. I did a lot of traveling in the early days of my career, and clearly family took second place. And so I'm grateful that my wife had the patience and my children had the good sense to know which side the bread was buttered on. And so I was allowed to advance my career at their expense. So I'm grateful for that as well. But the entire culture at Wisconsin over the years has been one of great support. I've served under either 5 or 6 deans now, all the deans at Wisconsin, and I was always encouraged. When I was first hired, the dean, Arthur Ewell, who was a very interesting man in his own right, when I met with him for the first time to talk about doing work on the faculty, he gave me some advice. He said, "I'd like you to do a good job in teaching, but I want you to do an excellent job in research." So it told me that the orientation at Wisconsin would be not only a recognition, but an assessment of your research contributions for promotion and for other aspects. And I never forgot that as an introduction to faculty life. And it's turned out to be pretty accurate as the driving force for promotion.
Well, you've certainly achieved in both areas. I might say that the scholarly effort of being both an educator and a researcher, you have a great legacy because, in a sense, some of the outstanding researchers that you helped train have in turn developed to be those kind of teacher researchers themselves. And so that should be a degree of satisfaction to you. I remember a specific meeting you and I had in that upper penthouse on the 5th floor that you had in the old pharmacy building, probably back in the 70s, where I was talking to you about adhesion. And you were talking to me about bioadhesion. And we both were fascinated by the fact that neither of these disciplines, I was working in microencapsulation and coating, and we couldn't measure adhesiveness. I don't know if you remember this conversation.
Yes, I do, Art.
It was over an hour and a half, even though I had planned to just come by and say hello. And you were telling me about how to measure, and I was telling you that the scientists and physical scientists had a unit called a TAC, which was a CGS system, 1 dyne per square centimeter, unit of force to pull apart adhesion. I said, we should be doing this in pharmaceutics. And you said, we should be doing this in bioadhesion. And I said, wow. Now, you went from there to looking at mucin and the whole area. This is a fantastic story, Joe, about taking the seeds from one area and breeding it into another, and that led to the work you did in vaginal drug delivery and oral drug delivery and the role of the esophagus. Tell us about that, how you got into bioadhesives.
It was actually as a result of one of my graduate students. I think, to be fair, most of the advances I've made in my career have been as a result of having really smart graduate students and postdocs. This particular graduate student, his name is Kanam Park. He's a faculty member at Purdue, and his wife was one of my students as well, Haisun Park. But he came to me one day and he said, you know, it'd be nice if this polymer that we were looking at would stick to tissue. And I thought, why didn't I think of that? And that became the beginning studies. That polymer subsequently became commercialized in a lot of vaginal products. And the importance of it is, and I can tell you just with one example, if you introduced a solution into the vagina, its contact time would be about 50 or 60 minutes. Whereas if I introduce it with a bioadhesive, it'll be there for 4 days. And so you can treat local conditions very effectively. You can improve the absorption of drugs enormously. And when you think about other areas, the eye, the contact time of a solution that you put in your eye is about 15 to 30 seconds, and then it's washed away. Imagine being able to stay there for a day and not interfere with vision. And so there were a lot of things that could be done, and we've really only just touched the surface of the application of this. But lots of people have now pursued this area, and it's becoming prominent in a lot of different ways. And I'm thrilled by its utility, not just for the polymers that I've worked with, but for others as well. So it's turned out to be a pretty meaty area, and it's been very satisfying art. Yeah.
Well, I remember from those early discussions and how you, in a sense, opened up a new approach to a traditional area that had been underserved, perhaps, in drug delivery, to following up maybe almost a decade later, where you and I were together on a Conrad program at NIH, if you recall, speaking, and you were identified at that time as giving the keynote talk on vaginal drug delivery. So by that time, I think that you paved the way for a remarkable advent of new pharmaceutical products in that arena.
Well, interestingly enough, the world of fertility and contraception is still not well served from a drug delivery point of view. I think in part because women's healthcare products have been stepchildren. You know, you can think of one pharmaceutical company that specialized in that. That's Ortho Pharmaceuticals, a division of J&J. But outside of that, you're hard-pressed to think of other companies that had focused research efforts in this area. But there's a growing recognition that women are the purchasers, and women health care is at least on an equal footing with male health care. And a lot of these issues, although specialized, are deserving of much more research efforts than have been put in the past. And it's starting, but it's a very early stage to get the attention that it really deserves.
You're quite right, and of course that has changed, and probably not to where it really should be in order to have equal gender footing. But I think mainly because of contributions that you made in that vaginal drug delivery area. One of the areas that you contributed to, not just in vaginal, but in other areas such as ophthalmic drug delivery, is the recognition that in vivo correlations with cell tissue studies and organ studies and animal work would allow for some investigative work where maybe women could not fit into the clinical picture. But then you identified, of course, that paucity of women in the clinical portfolio.
Right.
Would you talk a little bit about the cell studies and the cell culture studies and the tissue studies, and how valuable were they in predicting success in drug parameters such as, the pharmacokinetics of drug delivery and maybe even the selectivity and targeting of drug delivery?
Yeah, before I do that, I would just go back to vaginal drug delivery and tell you just a lighter story. My wife refused to attend any lectures that I was giving on vaginal drug delivery.
Because she thought you weren't the expert, or was that the...
She thought I was the expert and wanted to know why I was the expert.
Ah, I see.
So that became an interesting aside.
Yeah. Excuse me, but it is true that almost all the experts in vaginal drug delivery today are still men.
Oh, yes, without question. Yes, absolutely.
Mm-hmm.
I think that's changing a little bit. There's a lot more women who are going into that field because other women are much more comfortable seeing a female. Mm-hmm. But it's still underrepresented, actually.
Right.
The issue of animal models is an absolutely intriguing one, as you well know, Art. Everybody would prefer to use an animal as a surrogate for the human. It is difficult often to get humans to volunteer, especially for basic science studies. And in fact, in the old days, we could use prisoners as paid prisoners for some of this work, but never really the basic sciences. That was much too difficult to do. And so everybody looked for ways to model human behavior and human disposition of drug delivery systems. And with the advent of cell cultures, which were far less expensive than animals per se, you're getting rather remote from the large animal. And often in cell cultures, you're so remote that you can't predict behavior. And to this very day, I think it's fair to say that no animal model is satisfactory to emulate human behavior and human processing of drugs. It is a serious disadvantage from a speed of development point of view, but toxicity issues that manifest themselves or don't manifest themselves in animals, you can't apply that to humans, so there's no extrapolation. And it is almost on you have to do it in humans to actually make a comment about human behavior and human processing of this drug. I don't know if we'll ever get to a point where we'll be able to use an animal model for human behavior. I remember we have had all kinds of studies and programs on the oral route of administration, and the question always came up, is the mini pig or pig better than a dog, which in turn is better than a rhesus monkey, and on and on and on and on. There is really no adjudication of that issue. It was left just you have to do humans if you really want to know human behavior. It's unfortunate, but that's where I think we're at right now. But there's improvements being made every day. It's just that I don't think we've solved that problem yet.
There were and have been a lot of contributions that came out of your laboratories in the development of analytical instrumentation and approaches that were unique for monitoring the efficiency or the specificity of drug delivery.
Right.
What stories could you tell that led you to some of the kind of contributions that you became well known for in looking at, for example, the use of radioisotopic tracing and so forth, in identifying drug delivery efficiency and design?
Well, that was that and probably every other advance that I published on was out of necessity. If you think about the eye, you're dealing with levels that are not detectable by conventional means. And so I was forced to use something else. And there's something else turned out to be technetium tracer to look at tear turnover time and tear flow rates, and it was a remarkably efficient tool for my purposes. You could get real-time results and in a short order and fairly inexpensive, and I could often do this stuff in humans because technetium was safe for use in humans. And that, among other things, some other analytical issues that I dealt with, was really out of necessity. I was forced because of the low levels I was dealing with trying something else. And it worked. But I can't take credit for a lot of these things. Oftentimes it was the graduate student or postdoc who, when confronted with this, would come back and say, we ought to try this. And I was more than happy to try that. So much of the credit really goes to the very good students and postdocs I had over the years.
You've contributed in your role as a researcher, as a mentor to students in your research programs, as a academician, but you also contributed in many ways as a volunteer for a lot of professional societies and associations. You spoke of the Control Release Society, but you played a major role in the pharmaceutical societies, the various organizations. And you played a leadership role, elected office, and you were pivotal in some of the major issues that reflected on that. In a career reflection, how would you, let's say, focusing primarily on the pharmaceutical disciplines now and not the chemical or control release areas, how would you reflect on your satisfaction in that leadership role? And you were, as I recall, pulled into some pretty tough assignments in terms of provocative periods of controversy.
I think partly it has to do with a sense of an obligation on the part of scientists in general and pharmaceutical scientists in particular to serve on public panels that are advancing the frontier of our discipline. I served for more than 20 years on the United States Pharmacopoeia as an elected member. And I always thought that while time-consuming, it was a standard-setting agency that would benefit everybody if it had proper input. And I, along with hundreds, if not thousands, of other volunteers, did exactly the same thing. And so I always thought it was part of the mission of especially an academic who is perceived to have more time than an industrial scientist, would volunteer for these types of activities. I served on a lot of different panels, but I always felt that it was part of an obligation to do that, and I never begrudged that use of my time because I thought it was helpful in advancing. And in the USP, sometimes you would make a suggestion and a recommendation and it would pass, and sometimes it would fail. But you always had an opportunity to put your oar in the water. And that was very important for me, that I could be heard on issues that were important to me. The definitions, for example, of controlled release items, and the various in vitro studies to characterize these, that all came out of activity in the USP. Would it have come out if I didn't serve? Probably. Would it have taken a bit longer? Probably as well. But I was heard, and I was pleased to participate. It was in my interest to participate, and it was an obligation, I think.
Well, it's exactly that role that I envisioned you elaborating on. The other thing that happened about that time, Joe, which I'd like you to comment on, is that the USP's leadership and the emergence of global issues and standardization led to a lot of councils on harmonization, which you had a leadership role in.
Yeah. And
trying to get the world to see a level playing field, not just in definitions, but in standards of quality and performance.
Yeah, I was instrumental, but not the major player in this world of harmonization. But it became very important for countries in the EU, outside of the EU, and Japan and emerging countries to have standards that they could apply for their pharmaceutical products. And the USP really was a pivotal organization. It was the lead organization, in my view, for this issue of harmonization. And each country looked at its pharmaceutical supply in a little different way. The Japanese, for example, were more interested in safety of their products than they were in either manufacture or therapeutic benefit. That's a cultural issue on their part. But some standard that looks at across the board and says this is the way you should assess these products, and this is the way we will deal with it in the future, has led to a lot of very useful things, especially in the world of controlled release. You can make a product controlled by having what is called a burst effect, where you get a lot of drug going into the system immediately. Of course, you'll kill a lot of patients in the process. And some recognition by the regulatory agency that works hand in glove with the USP is important. And that's one of the issues that I think has come as a result of a better appreciation of how these things are supposed to function. And so harmonization is clearly a very important issue. The FDA is under a lot of fire to approve drugs quickly, and it's a serious problem. You know, I think the World Health Organization classifies that there is something in excess of several thousand diseases. We effectively only treat 300 or 400 of them because there's no money to be made in these other diseases. And so the capitalist system lends itself to profit making and speed of approval. And of course there comes danger with every one of those things, because that's when you let your standards slip and approve without doing all of the homework. And the enormous number of lawsuits that we see in class action suits today, I think, are as a result of trying to move too quickly. But if you slow down the FDA, there will be a hue and cry from all of the industry saying you're going too slow. So it's a no-win situation in a sense. Yeah. But I do entirely love the approach of harmonization. It works very slowly from my point of view, but I think it takes time to really encompass all of the different issues that the various countries of the world are looking at. And the pharmacopoeia is a perfect way to deal with harmonization.
In addition to your interest in harmonizing and global outreach, one of the major contributions, and we can cite in your career, is not only as a prolific publisher of science, but as a contributor to journals in the editorial capacity. You have given much service in journal subscription, dissemination of science on a global aspect and so forth. Could you talk to us about your role in editing and contributing as a reviewer initially, and then in your editing roles? You've had many editorships of journals, and you continue to do that today.
Yeah, once more, it's an obligation that scientists have to review other people's work, either for publication or for grants. And it's a non-salaried activity, and it's subject to considerable abuse. It's an anonymous system largely, but occasionally it slips out who did the review. And it's unpleasant when you have to turn down a scientist's article for publication. On the other hand, you tell yourself that you're saving them from larger embarrassment by turning it down. And that usually satisfies us. But I know that, well, I'll tell you a story. The very first article I published in the ophthalmic world was reviewed by a physical chemist who was at UC Berkeley. And it was a punishing review, Art. He wanted me to correct virtually every dot and comma in this manuscript. And I think it took the better part of a year to get this reviewed. It was ultimately published. It turns out to be the best manuscript I ever published. But at the time, I could have killed the guy, because every time I'd go back to the journal, they'd say, well, we've got to send it out once more for review. And it was always the same guy. I met him years later, and he told me he liked the final product because he had such a major input into it. Yeah. But I thought that was an obligation that good scientists have to prevent embarrassment by others and to prevent misleading science from getting into the literature.
You know, I wanted to cover one point when you talked about this. In addition to your role as a contributor, you're also well known as being a talent of sorts, not only as a musician, but as a humorist. Could you share with us, as we close our discussion today, your adventures in that area?
Well, I've never really pushed the humorist angle in my life. I think I'm just a cynic based on background. But the music part of it, I thoroughly enjoy as a compliment to my career. My colleague Ken Connors took up the banjo, and we formed the Henderson Hasselbach String Band years ago. And we had a very prominent cast of characters. George Agraffi was in there, and Glenn Sonnedecker was in there, and we had very good people. And of course, they played strange instruments, the jugs, the washboard, a variety of things. But we volunteered for talent shows whenever we could, and we'd often suggest that we would be good if they invited us. So it was sort of fun. And I look back at those years and the photos that I see of these pictures, of these events, and it gives me great pleasure. Real fun.
Well, on that note, I want to thank you, Joe, for appearing on this series.
My pleasure, Art.
It's been a pleasure talking with Joe Robinson, Professor Robinson from the University of Wisconsin. Thank you very much, Joe.
Thank you, Art.